Okay. Hi, my name is Rick Pazdur, and when you were mentioning that we've been there since 1999, I'll just start with a story. On my first day at the FDA, I had just come from MD Anderson. I was a full professor there and a tenured full professor, and when I took the job at the FDA, people said, Rick, how can you give this up and go to the FDA? You must be nuts, okay? And I just realized the potential that existed at the FDA really to make a dramatic change in drug development in oncology, and I've been very happy. But at the time that I got there, I was addressing the staff. And when I addressed the staff, Bob Temple was sitting next to me. I don't know if many of you know, but he's kind of an icon of drug development at the FDA. He was my office director. And I said, I don't believe that anyone should work for government for more than 2 to 3 years, and then they need to leave. Well, he just gave me this look like you couldn't believe. And it's interesting that I didn't take my own advice. I remained there and continue to remain there, but it's an interesting place to work. I want to make a disclosure. First of all, I know nothing about TB, okay, nothing, okay? The last patient I treated with TB was probably in 1973 or something like that. So I hope the field has changed, and I have not kept up with it. You're laughing, that's troublesome. Still using ethambutol or whatever it is, and INH or what all those drugs were. So I know nothing about the disease, nothing, okay? My knowledge stopped like in 1973. And the other thing that I'd like to bring up is I think there are some limitations on the extrapolation of the two disciplines. Needless to say, let me kind of give you a story. First of all, what percentage of drugs that are oncology drugs, if you take a look at the whole world of drug development, what percent is oncology? 10%? More people want to raise their hand? 10%? 20%? 30%? 40%? 50%? You guys are way over optimistic. It's about 40% of all new drugs are being developed in the area of oncology, and that includes malignant hematology and oncology. And this is a really active area for the pharmaceutical companies because you have to take a look at the total kind of environment of we're looking at drugs in cardiovascular areas, psychiatric diseases, neurological diseases, infectious diseases, antivirals, pain medications, et cetera. But when you take a look at almost all of the new molecular entities, about 40% of these are in the area of oncology. So it really represents a major investment by the pharmaceutical industry. And about 40% or 50% of our breakthrough designations, which we'll be happy to talk about, are in that area. I want to go back to where we were when I began the field. And I began oncology in about 1979, so you could guess my age from that. And to give you an idea of what was going on in oncology at this time, I was living in Chicago and had grown up in Chicago. And at that time, oncology was really in its very early stages as an academic discipline. In fact, when I took the boards in oncology, it was only the second time that they actually offered a separate board in medical oncology, distinct from hematology. In my medical school, I remember the pharmacology lecturer saying how lucky we were to have a medical oncologist on the whole staff of the entire medical school. The largest program in Chicago, which was at Rush Presbyterian St. Luke's on Harrison Street there by Cook County Hospital, had only 8 medical oncologists, and that was the largest medical oncology program. Switch now, Loyola University, where I went to medical school, has about 40 or 50 medical oncologists, has a huge cancer center, has a program that is involved with a lot of drug development, phase 1 studies, phase 2 studies, phase 3 studies. So the field has rapidly changed. When I went into medical oncology in 1979, if one took a look at all of the oncology drugs that we had, how many do you think there were? There were about 35 oncology drugs, and that included drugs that really weren't even kind of oncology drugs. They were like helatestin that you could treat, you know, various breast cancers for. They were still doing oophorectomies and hypophysectomies for women with breast cancer. They counted prednisone as an oncology drug and salumedrol as an oncology drug. How many drugs do we approve in the last year? New molecular entities, to give you an idea. So we had 35 total drugs then. Last year, we approved about 15 new molecular entities in oncology. The year before that, we approved 15 new molecular entities. The year before that, we approved about 12 to 15 new molecular entities. So the field, as you can see, has really escalated in that area. When I started oncology, okay, in 1979, and I hope I'm not boring people by this, okay? Okay. I'll ask you, what was my impression of what oncology would be? Well, first of all, I went into oncology because it was a very, I would say, investigational area, okay? It was very academic, and I was very interested in academic medicine as my career kind of testified, subsequent career testified to that statement. And so I believed that all of medical oncology would be practiced in tertiary medical centers, that there would be really no role for community oncology because all of these therapies were toxic, they were relatively ineffective, there were only a few diseases that we were actually curing that could be counted on one hand basically with chemotherapy. So I thought most of the drugs and most of the care of cancer patients would be conducted in the academic center. Was I right or wrong? Wrong, okay, dead wrong, okay. The vast majority of people in the United States are treated in the community, well over 80% of them. So it's a market difference and a market departure from what I thought the field would be. Referencing to drug development, I thought that most of the drug development in the area would occur by the government entities, specifically the NCI, the NIH, right? It was a relatively new area. This is what government should be doing. I felt that the pharmaceutical industries would have very little interest in oncology. Why? Well, you know, these drugs were really very toxic, classical cytotoxic drugs. Okay, they had a tinge associated with the field of oncology, especially in the 1970s before Betty Ford and other personalities really emphasized a more front face of oncology, was associated with kind of death and dying, and many pharmaceutical companies might not want to be have a kind of stigma attached to specializing in kind of a field where most of the patients were going to be dying. There probably would be little market for these drugs because unlike antibiotics, where these drugs are used perhaps on a more chronic basis, or other antihypertensives or cardiac drugs that are used in antihypertensives, these oncology drugs, for the most part, because of their toxicities, were going to be limited to you know, a very short course of therapy. So my second hypothesis that all the drugs in oncology would be coming from the NIH, was that a correct analogy? No, okay, so nix that area. What happened, obviously, is that drug companies recognized that there was a market in oncology, that they could charge for this, and really they grew with the discipline of medical oncology, okay? being in the field for this past almost 40 years, I've seen many ups and downs. And for the first part of my career, there was basically the era of conventional cytotoxic chemotherapy, where you're all accustomed to these drugs. They were very similar to Cytoxan, Taxotere, the anthracyclines and modifications of these. These were drugs that had relatively modest activity. They were used in combinations. They had pretty severe toxicities associated with them. We managed them, and in fact, my late wife said that, Rick, the only reason that medical oncology exists as a specialty is for the management of toxicities. You know, if these drugs were relatively non-toxic, the surgeons would be very willing to give them out, and radiation therapists would also. But these drugs had such toxicities associated with it, they really needed the support of internal medicine specialties that could manage neutropenic fevers, severe mucositis, dehydration that required central lines, et cetera. So the first part of my career was involved with drug development in the cytotoxic area. Then what happened was the development of imatinib for the treatment of CML, which kind of changed the course of medical oncology because that was truly the first targeted therapy. And subsequent targeted therapies emerged, tyrosine kinase inhibitors. We started about 10 years ago looking at molecular pathways of drugs. We have specific drugs that target specific pathways, for example, the BRAF drugs, the MEK inhibitors for the treatment of melanoma, which has transformed these diseases. And then most recently, we have developed the rebirth of the area of tumor immunology, particularly looking at checkpoint inhibitors. And I won't go into the science of this. I'll just tell you, for those of you that are unfamiliar with this whole area of checkpoint inhibitors, these include drugs such as nivolumab, and you could see that on television commercials that give this, I think, overly optimistic picture of lung cancer. Nobody wants to have lung cancer, but it seems that these advertisements kind of go way overboard in promising results. But these basically take the brakes off of your own immune system and cause the immune system to rev up against these tumors specifically. This required some retooling of how we do clinical trials. First of all, we started taking a look at single-arm trials in very refractory patients. So these were patients that had really received all of the standard therapies. And we started treating these patients with, you know, a drug right out of phase 1 studies, since we knew the dose. And we got a response rate, and response rates for the most part is just tumor shrinkage. And they have to be maintained for a certain period of time. And that provided then a reason to approve the drug under accelerated approval on the basis of a response rate. So the response rates could be 14%, 15%, 20%, 30%, indicating that about 20 to 15%, whatever the point estimate would be of those studies, would be indicative of patients that have gotten a tumor reduction. So the tumor had to reduce by a certain pre-specified size limit. In addition to that, we started to take a look in randomized trials at different endpoints other than overall survival, because overall survival, and I'll be happy to talk about that, was becoming a much more difficult endpoint to measure. And that had to do with looking at progression-free survival, which really measures the differences in a randomized study between the progression of patients and when they progress or die, but primarily radiographic progression of diseases. And we were able to approve many drugs on those basis. So that really heralded in a lot of interest by the pharmaceutical companies by looking at alternative endpoints other than overall survival to get promising drugs on the market. When I first came to the agency in 1999, the dogma, in fact, I was one of the reasons I came to the agency. I was actually one of the investigators on a specific drug in oncology and developed the drug from phase 1 studies, phase 2 studies, phase 3 studies to a adjuvant therapy. Unfortunately, that drug never got approved, so I'm one of the only people in the FDA that actually knows what it's like to be on the other side of the arrow, so to speak. But the dogma at that time was that you needed 2 randomized trials which showed a survival advantage. End of discussion. End of discussion. 2 trials, 2 randomized trials. I don't care if there are 1,000 patients. I don't care if there are 1,000,000 patients. You need 2 randomized trials that show a survival advantage. End of discussion. Well, that probably was not going to promote drug development. First of all, oncology is a bit different, and I don't know how this relates to the area of TB, but you know, it's hard once you do, because of the kind of small nature of these diseases that we're dealing with. We're not talking about developing a drug for an antihypertensive or a congestive heart failure drugs. Even if you take a look at refractory breast cancer, it's only a relatively several thousands of patients that you could enter on a clinical trial here. Pharmaceutical companies did 1 trial to kind of manage their risk and get an idea of how the drug worked, either at an improvement in overall survival, response rate, whatever. But then it became very difficult after you got the results of 1 trial and it showed success in a situation where you had no other therapies to really demand another trial. So that led us to really accept 1 trial, and I can't tell you the last time I saw 2 randomized trials coming in for an initial approval in oncology. It's a rare event that we see 2, and we just have to eat it and live with it. And I think basically that has promoted a lot of interest because it minimizes the risk in the pharmaceutical world, which their major issue is risk reduction and how to control risk in drug development. One of the other things that we most recently did, especially with the advent of these new immunological agents, which I think really hold the promise, if they do continue and other generations continue really to transform the area of medical oncology, is come up with what we call seamless drug development. And you're all obviously very well aware of the traditional drug development scheme of phase 1, phase 2, phase 3 studies. I'm not quite sure if this occurs outside of the oncology world, but what we piloted with some of these immunological therapies is rather than stopping in these distinct areas of drug development, sponsors would go into a phase 1 study, then they could go into a phase 2 study seamlessly without stopping the trial in a particular disease, in another disease, in another disease, looking at a drug combination, looking at it in pediatric patients. So there wasn't this stop, start, stop, start, stop, start of these relatively artificial areas of drug development, and that has really escalated, you know, the development of these drugs. Now, one of the problems that we have in oncology is the issue of the herd mentality, and here again, this has to do with the reduction of risk by the pharmaceutical companies. And for example, the PD-1 drugs, we all feel and we all know that it's better to have more than 1 drug of the same kind of class on the market than just having 1 drug, because there could be drug shortages, et cetera. However, what has happened in oncology many times is you have this herd mentality where, yes, a couple of drugs are good, but you don't need 20 of the same drug. Okay, and that's what we're seeing with many of these companies, is that there's a herd mentality to coming into the area, which poses some difficulties. It certainly doesn't promote innovation to just keep on doing the same drug over and over again, but it does reduce their risk as far as whether or not the drug should be approved, since they really could follow kind of the paradigm of how a previous company had done their drug development. So I'm going to shut up now, okay, and let Ed take over. But I hope I gave you kind of a glimpse of kind of where oncology has gone in 40 years. What has been very rewarding to me as a clinician and then as a drug regulator is to see diseases where there were absolutely no therapies or marginal therapies, and seeing really very promising drugs and very promising things come out, and also them to be advanced, these promising therapies, to minimal disease patients where there is an option for cure, for example, an adjuvant therapy. Some examples of that, as I mentioned, CML, where the standard therapy when I began was hydroxyurea, basically, and to just artificially just control the counts. And now we have survivals in that disease that really age-match controls without the disease. Other areas include myeloma, where the only drug when I started out, a regimen was melphalan and prednisone, and now we have a plethora of drugs and a marked improvement because of all of these drugs. And they didn't individually show survival advantage. It was getting drugs out there that show evidence of biological activity and then putting them together or using them sequentially that became very important. Other areas, renal cell cancer has been transformed by the tyrosine kinase inhibitors. The area of melanoma, skin cancer, all you have to do is take a look at Jimmy Carter. When I originally heard his disease with the presence of brain metastases, I said, well, this guy's a goner, but with the advent of targeted therapies and PD-1 therapies, these diseases can be transformed. I have a friend that lives here in Washington who had brain metastases, pulmonary metastases, liver metastases, went on targeted therapies, the BRAF inhibitors and MEK therapies, and went into a CR. And now the debate is, after 5 years, is should she stop these drugs? How long does one continue these? Now here again, that represents kind of the unusual, but I can't tell you how many patients that I had when I was treating patients in Detroit in the 1980s and at MD Anderson in the 90s with these diseases that, I just were signing death certificates after death certificates after death certificates on them. So I hope this gave you a glimpse of what we do in oncology, and I hope we could perhaps extrapolate some of this to the area of TB.